red blood cells from chickens are used in the hemagglutination test to detect avian influenza viruses, and in the hemagglutination inhibition test to detect for the presence of subtype-specific antibodies. Collect the blood with an anticoagulant, such as Elsevier solution, used in a ratio of 1:1. Prepare the correct volume of anticoagulant in the syringe before the blood is taken. Note that acid citrate dextrose in the proportion of 1 part to 3 parts blood is also used. Chickens used for the supply of blood should be specific pathogen-free or AI-specific antibody negative, and should be housed separately from chickens used for other purposes. More than one chicken should be bled and the blood should be pooled. To carry out hemagglutination tests and hemagglutination inhibition tests, preparation of 1% red blood cell suspensions are required. First, prepare a 0.05% bovine serum albumin solution in phosphate buffered saline. Bovine serum albumin is a nutrient for cells and extends the shelf life of the red blood cell suspension. With the help of a graduated cylinder, measure the correct volume of the prepared solution based on the final amount of red blood cell suspension required. Place it in a sterile glass bottle and proceed with the red blood cells washing procedure. Remove the needle from the syringe and pour the contents into a container suitable for centrifugation. Remember to treat the blood gently to avoid hemolysis. Centrifuge the blood suspension at 1500 to 2000 G for 10 minutes. After centrifugation, the cells form a pellet. Discard the supernatant. Fill the container with PBS to wash the red blood cells and mix by gentle inversion. Centrifuge at 1500 to 2000 G for 10 minutes. Repeat this process 3 or 4 times until the supernatant is clear. After the wash, remove the supernatant using a pipette, being careful not to disturb the pellet of red blood cells. Now prepare the red blood cell suspension at the desired concentration by resuspending the appropriate volume of pelleted cells in a 0.05% bovine serum albumin PBS solution. The accuracy of the red blood cell percentage has to be controlled by a spectrophotometer with the help of a cell counting chamber or a microhematocrit. The ratio of the serum to red blood cells may dramatically affect the sensitivity of the agglutination tests. A cell suspension that is too concentrated or not concentrated enough may produce false positive or false negative results. Prepare an additional red blood cell suspension at a concentration of 10% for the pre-treatment of sera from species other than chicken. Again, add the necessary volume of pelleted blood cells to the 0.05% bovine serum albumin PBS solution previously prepared. Store the suspension at 4 degrees for up to 5 days. As the cells settle under gravity, control that the storage solution remains clear. Any red colour in the storage solution indicates hemolysis of the cells, and in this case a fresh suspension of red blood cells should be prepared. The World Organization for Animal Health recommends the HI test as a diagnostic standard test for avian influenza serotyping. Perform the hemagglutination test and the hemagglutination inhibition test on V-bottomed 96 well plastic microplates. The reagents required for these tests are reference antigens and sera, phosphate buffered saline of pH 7 to 7.4, PBS plus albumin, and 1% red blood cell suspensions. Note that lyophilized antigens and sera should be stored at -20 degrees Celsius. Once antigens are reconstituted, store them at -80 degrees Celsius, while reconstituted sera for positive and negative tests can be stored at -20 degrees Celsius. Keeping reagents on ice when they are out of the refrigerator is strongly recommended. Determining the titer of the reference antigen by means of the hemagglutination test is required each time the antigen solution is prepared. Reconstitute the lyophilized antigen with 1 ml of distilled water. Mark the microtiter plates with the subtype of the antigen in use. Distribute 25 microliters of phosphate buffered saline into each well of the first row for antigen titration and of the second row for red blood cell control. Dispense 25 microliters of antigen in the first well of the first row and make two-fold dilutions across the microplate. Discard the last 25 microliters. Add 25 microliters of PBS to all wells. Gently shake the red blood cell suspension to mix the solution and prevent pelleting of the cells. Finally, add 25 microliters of 1% red blood cells to each well. Mix by tapping the microtiter plate gently and incubate it for 30 minutes at 20 degrees Celsius or 60 minutes at 4 degrees Celsius. Read results when the red blood cells of the control wells have settled, forming a distinct pellet in the bottom of the well. Reading is done by tilting the plate and observing the presence or absence of a tear-shaped streaming of the red blood cells. Wells with no hemagglutination should flow like the red blood cell control wells. When the tear-shaped streaming in the red blood cell control wells is complete, mark the highest dilution giving complete hemagglutination of the red blood cells in the first row. This dilution contains 1 hemagglutinating unit, HAU. Record the information on the tube with a permanent marker. To perform the HI test, a 4 hemagglutinating unit antigen solution is conventionally used. In some laboratories, an 8 hemagglutinating unit antigen solution is preferred. To prepare a 4 hemagglutinating unit antigen solution, calculate the total amount of antigen solution required to test all sera under examination. Calculate the hemagglutinating titer containing 4 hemagglutinating units by dividing the antigen titer by 4. For example, if the hemagglutinating titer is 1 in 512, 4 hemagglutinating units will be obtained by dividing the titer by 4. That is 1 in 128. A dilution of 1 in 128 of the hemagglutinating antigen will be used to prepare the antigen solution for the HI test. Prepare the antigen solution according to the calculation. Non-specific inhibitors present in serum samples collected from species other than chicken, such as mallards, quails and others, may cause non-specific cross-reactions in the HI test. Such substances can be removed by pre-treating the serum by heat decomplementation and by adsorption with chicken red blood cells. Heat the serum in a water bath at 56 degrees Celsius for 30 minutes before pre-treatment with a 10% red blood cell suspension. After decomplementation, add the serum to the red blood cells in the ratio of 1:1. Allow absorption of non-specific inhibitors for 30 minutes at 20 degrees Celsius. From time to time, mix by gentle inversion to allow all inhibitors to come in contact with the chicken red blood cells. Red blood cells are then pelleted by centrifugation at 1500 to 2000 Gs for a few minutes. Serum is now ready to be used in the HI test. The World Organization for Animal Health recommends the hemagglutination inhibition test as a standard test for avian influenza subtype-specific antibody detection. In surveillance campaigns, it is recommended to perform serological diagnosis of both OIE notifiable avian influenza H7 and H5 viruses. Identify the samples under examination on the microtiter plate with an indelible marker. The last three rows will be used for the positive control serum, the negative control serum, the control of the 4-HAU antigen solution, and the red blood cell control respectively. Mark the division between the 6th and the 7th well of the last row and use the first 6 wells for the back titration of the antigen solution and the last 6 for the red blood cell control. Distribute 25 microliters of PBS in all wells of the microtiter plate, with the exception of the first well of the antigen solution control wells in row H1. Dispense 25 microliters of serum into the first wells of the first column, using a new tip for each sample. After the addition of the serum samples, add 25 microliters of the positive control serum and 25 microliters of the negative control serum in the next 2 rows. It is recommended to include reference control sera for each batch of samples under examination. Using a multi-channel micropipette, make 2-fold dilutions of the sera across the plate and discard the last 25 microliters. Add 25 microliters of the relevant antigen suspension containing 4-HAU across the plate, with the exception of the last row. To confirm the presence of the 4-HAU in the antigen solution, dispense 25 microliters of the solution in the first 2 wells of the antigen control row. Remember that in each microtiter plate, the last row is left for the back titration of the antigen solution and for the red blood cell control, making 2-fold dilutions from the 2nd well to the 6th well. This will give 4-HAU in the first well, 2 in the 2nd, 1 in the 3rd, 0.5 in the 4th, 0.25 in the 5th, and 0.125 in the 6th. Discard the last 25 microliters. Dispense 25 microliters of PBS with the addition of albumin if preferred in all wells of the last row to bring the volume of the wells to 75 microliters. Mix by gentle tapping and incubate the plate at between 20 and 24 degrees Celsius for 30 minutes or for 60 minutes at 4 degrees Celsius. Finally, add 25 microliters of 1% red blood cell suspension in all wells of the plate. Mix by gentle tapping and incubate the plate at 20 to 24 degrees for 30 minutes or 60 minutes at 4 degrees Celsius. Results are read when the red blood cell control has settled. This is done by tilting the plate and recording the presence or absence of tear-shaped streaming of the red blood cells at the same rate as the control wells containing red blood cells alone. The HI titer is the highest dilution of serum, causing complete inhibition of the hemagglutination. The test is valid when the negative control serum has a titer of less than 1 in 8 against the 4-HAU antigen solution. The titer of the positive control serum should coincide with that declared. Note that 1 dilution of difference is permitted. The 4-HAU control should show complete hemagglutination in the first 3 wells containing 4, 2 and 1 HAU respectively. HI titers may be regarded as positive if there is inhibition at a serum dilution of 1 in 16 or more against 4 HA units of antigen.